Morning.
Good morning.
Loud kicking. How you doing, Evan?
Good. Happy New Year, I guess.
New Year, yeah. Morning, Morgan.
Morning.
Good morning, good morning. Welcome back. Happy 21. Morning to those just coming in. Happy New Year. Welcome back. Still showing about 12 people not here, so waiting just a moment. Okay. Give it about 30 seconds and then we'll go. Okay, I've got to submit attendance here, so I'm just double checking. Drake, if you're here, identify yourself, either speak or go on the chat. Give me just a second for the attendance. School internet has been real touchy. I don't know if anybody else is experiencing that. Okay, Edward's here. Eddie's here. Okay, got that. Bear with me. One more time, Drake, anybody, if you're here, speak up. Catherine, if you're here, speak up or chat. Marissa. Alexander Owens, are you present? I see you just came in now. Okay, you're good, Alexander. Aiden, are you here? Chatter, speak up if you're present, Aiden. Okie doke. All right, anyway, welcome back. Happy 2021. I hope you got to do something the last couple of weeks that allowed you to sort of recharge your batteries and refresh yourself a little bit. We got 2 weeks to wrap up the semester. Again, as you probably already know, we're not doing a traditional semester exam. Not this Thursday, but next Thursday, which is the 14th, we'll probably do our unit test on cells. That'll be our last test for the first semester. And then the next week we start second semester. Currently the plan, last I heard, the plan is still that we will come back in person. I unfortunately can't tell you what that's going to look like. We're still waiting information ourselves. So I'm assuming those that'll come back. There's been different discussions about whether we go back to regular 6 classes, whether we stay 3 a day, all that I don't know where the decision hasn't been.
Trev.
Yep.
So like, are we gonna go back to when we had like 5-minute classes and it was good?
I don't believe so. But I don't know.
Man,
I
like, do
don't
you know how
know.
long it's gonna be?
I do not know. I wish I could tell you. As soon as I know, I will send out a message to everybody and let you know exactly what's
All
going to
right,
happen.
sick. Thanks.
So anyway, all I know is second semester starts on the 18th. The plan is those that want to come back will come back. And I do not, unfortunately, know what either the in-person or the virtual student layout will be. But that's okay. We'll just roll with the punches and make it work best we can. The recent test scores and recent digital notebook. You're probably going, what is that lazy SOB done? Where the heck is my scores in my grade book? Why don't I see them? It was a 2-week break. I just didn't put them in yet. My own kids were home, so they will be in the next day or two, I assure you. And you'll have your most up-to-date grades, certainly no later than Wednesday. You'll see everything that should be there. Okay. Any questions about anything in general before we jump back in? Going once, twice. Okay, so today, if it go, second hour didn't run very smooth. It wasn't their fault. We had some glitches that I did not have first hour. I'm hoping we don't have them this hour. So if everything goes as I plan, we're going to spend about 30 minutes talking, me talking, about 10 to 12 minutes taking a look at a short video, and then the remaining time having you work in small groups on a task. So those are sort of the three things we're going to try to accomplish today. So to do the first part, you are going to want to go to Google Classroom. Is it showing up yet for you? Oh, here it comes. Okay. So you're going to go to Google Classroom under January 4th. You're going to want to open up this interactive notebook on cell membranes. It's a very short one. I didn't give you the whole notebook, but I gave you what you need for today. And if you'll open up this notebook, you'll see, like before, there's a few pages that are going to go along with a set of slides. It's only about 10 slides today, if even that many. I don't even know if it's that many. So you can go ahead and put your name on the book, and then we're going to be using these first couple pages in today's discussion to talk a little bit about cells. All right. So we give you a moment to get that opened up so it's ready to roll. And I'm going to jump between a PowerPoint and some stuff on the whiteboard. When I get to the whiteboard, you might want to have a sheet of lined paper or something of that sort to make a copy of the image. I suppose you can also take a screenshot out of it or a photo with your phone, but you might want to have access to the drawing that I'm going to do at sometime in the next half hour. All right, so let me share the PowerPoint. It's not even giving it to me. What's going on? Let me try this instead. It should be coming. It's not going to work. Sorry, small glitch. Give me one more try. All right, bear with me just for a moment. I'm gonna have to close this and try to reopen it. Wow. This thing is moving incredibly slow. All right, almost there, I think. So anyway, while I'm trying to get this up, before break, we were talking about cell structure and function. We made a contrast between prokaryote and eukaryotic cells. Prokaryotes are the earlier cells, the older cells. They have fewer bells and whistles. The analogy was it was like a studio or efficiency apartment. Everything was done in one space. But if you're only a single cell big, they work. They do the job, but they just don't have a lot of specialization. The prokaryotes, which are the more modern cells, which we are made of, they're more, the analogy was they're more like a mansion. They have organelles, they have a nucleus, they can multitask, they can do many different things at once. And we talked about that all cells have to be able to make energy, all cells have to be able to make proteins, and all cells have to be able to reproduce. And we talked about the different organelles that help do that. For like example, with energy, we talked about mitochondria and chloroplast and vesicles and lysosomes. For making proteins, we talked about DNA and the nucleus and the ribosome and the endoplasmic reticulum and the Golgi apparatus. For making more cells, we briefly talked about the nucleus, the DNA, and something called the centrioles. Those were sort of the things that we covered just before the break. Today, we're going to shift our focus to talking about specifically the cell membrane and cell transport, how cells move materials in and out. To get a cell, one of the first things that had to come about was the cell membrane, because the cell membrane is what makes the cell distinct from the rest of the environment. The cell membrane is what determines the internal environment from the external environment. And so we're going to look at the structure of the cell membrane today. And then on Wednesday and Friday, we'll look more at how the cell membrane carries out what's called transport of materials as we go. All right, so I think I might have this working. Let's find out. Oh, son of a gun, come on. Don't know why it's. All right, let me try it this way. Oh, there it is finally. All right, I think I might finally have it. Might. All right, looks like you can see it. It is. All right. All right, I think we got it. Let's get what we can out of this before it crashes. All right, so we're going to focus today on the cell membrane, particularly the structure of the cell membrane, and then how that is involved in controlling what goes in and out of cells. So let's move forward. So here we have a very basic cell. On this very basic cell, we have this outer membrane. All right, and then we have this blue interior, which represents the cytoplasm, and this dark circle, which represents the nucleus. So a very primitive cell. And so in order to have a cell, you have to first be able to create a distinct inside or internal environment and separate from the external environment. And so one of the primary jobs that the membrane does is it does just that. It separates the internal environment, the blue environment of the cell, from the outside white environment. And so you're separating it from its environment. Therefore, the cell membrane is often referred to as the boundary. It defines where the cell is. So this is the boundary of the cell. And what this boundary does is it allows the inside, this blue area, to be completely different if it needs to be from the outside white area. This blue area can have a totally different chemical makeup. It can have a totally different set of circumstances going on compared to the outside environment. And in order for this cell to survive, it has to constantly bring in materials, as we're trying to illustrate with the green arrow, and it has to constantly get rid of materials. You might see the slide occasionally blink. I can't get that to stop today, but it's not your computer, it's something on this end. So what are some of the things cells typically have to take in? Of course, some of the things they have to typically take in is food. And when we talk about food, we're talking about things like sugars, proteins, fats, your big macromolecules that we talked about before break. Those are things that a cell has to bring in on a regular basis in order to meet its energy needs and to have building materials. Besides that, it also needs salts, also needs oxygen, also needs water. These are all things that a cell has to bring in on a regular basis. Just like your body has to bring these in on a regular basis. Why does your body have to bring these in on a regular basis? Because your cells need it on a regular basis. That's why your body needs to bring it in on a regular basis. And I'll stop talking in a second so you can catch up on the typing. And then, of course, if you bring stuff in, you're going to end up creating waste products. And so you got to be able to get rid of stuff. So the red arrow is showing you that you have to be able to get rid of things when you don't want them anymore. So things have to be moved out. What types of things do we move out? We move out waste products like ammonia and salts and CO2 and water. And we also move out products like proteins that we make. These are just some examples of the things that cells move out. So I'm going to stop talking for about 30 seconds or so, in case you need to catch up on typing that in the box. You'll notice some of them are in both columns, like salts in both columns, waters in both columns, because sometimes you don't have enough salt or water. You have to bring that stuff in. Sometimes, depending on what you're doing or what you're eating or drinking, you have too much salt and water and you have to get rid of some of it. So some things waste product doesn't necessarily mean it's always bad. It's just waste product is something you have too much of. So in this case, water and CO2 or water and salts can be a waste product because you have too much of it. Where it can also be an intake because it's something that you do need. Whereas ammonia and CO2 are true waste products because if they accumulate, they can cause the body to go through some very harmful changes. Everybody okay? So that's the primary job of the membrane. The membrane sets the boundary, and then it's the membrane's job to make sure that these materials are moved in and out of the cell on a successful regular basis. All right, so cells need to bring materials in, and products or waste products need to be moved out. So how do you build a membrane? How do you create a barrier that keeps the watery contents of the cell separate from the watery environment? So you've got this dark blue interior environment that is water based. There's lots of water in there. Remember, cells are 70 to 90% water. So there's lots of water in here. But the environment, this light blue area, is also going to be water-based because living cells live in water-based environments. So you've got water outside the cell, you've got water inside the cell. So what might make a good barrier between these two areas where you have water inside and you have water outside? So you need something that does not interact well with water. In class, I might take more time to draw this out of you, but as you may recall, fats and lipids are things that do not mix well with water. Fats and lipids do not interact with water. If you put fats, lipids, oils in water, think about it, what do? They just sit there on the surface and float. They do not mix with the water. They do not dissolve in the water. All right. So remember, oil and water don't mix. So what we're going to see is this membrane, this boundary, is one of the main components is a lipid. One of the main components is a lipid, because lipids repel water, they don't attract water. And because lipids repel water, that's going to allow you to keep the two environments separate from each other. It's going to allow you to keep the two environments separate in the process. With me so far? Anybody out there? One person. All right. So let's take this little piece of the membrane. Ooh, look at that red circle with the arrow. We're going to blow that up and enlarge that and take a look at what that lipid structure looks like. So this is a blow up right here. I'm working on 3 different screens here. This right here is a blow up of that section of the membrane. And this is made up of a specialized lipid, and this is one of these enlarged. This is something called a phospholipid. Phospholipids are what make up the membranes of the cell. All right? So membranes are made of a special kind of lipid, and they are called phospholipids. They're called phospholipids because this yellow structure right here is a phosphate group. And these are 2 fatty acid tails, so phospholipid. Phospholipid. But this creates an interesting molecule. By having one part of it be a phosphate and the other part being fatty acid tails, it creates what we call a split personality molecule. It's got sort of a dual personality. One end of the molecule likes water, one end of the molecule hates water. And that creates an interesting chemical situation. So this is the phosphate head, where the yellow is there. And these are the lipid tails. Right? So got the phosphate head and we got these 2 lipid tails. That is a phospholipid. The phosphate end is attracted to water. And there's 2 different words we can use here to describe that. So the phosphate end is attracted to water. Phosphates have a charge. Water has a charge. They are attracted. They're attracted to each other. And therefore, we say that the phosphate is hydrophilic. You look at this word right over here, if I can get my cursor over there. Right here, hydrophilic. Right? Hydrophilic, hydro means water. hydrophilic comes from the word philia, which means love. So hydrophilic means it loves water. It likes to be with water. You can also call that polar. Polar means that it likes to engage with water. You'll see both terms used, hydrophilic and polar. Those both mean that you like water or you dissolve in water. Where the lipid is the opposite. Uh-oh. It's not showing up on my slide here. Let me see if I can slide that up. You bugger. Sorry, 1 second. Just going to try to move this so it can show up on the slide. Beautiful. It's not playing nice with the smart board. So anyway, the lipid end is repelled by water. It's what we call hydrophobic. Hydro, water, phobic from the word phobia, fear of water, does not like water. This is what we call nonpolar. Nonpolar means there is no charge. It cannot react with the water. So you have these two distinct regions. Phosphate likes water. It reacts with water. It dissolves in water. It's polar. The lipid end, hydrophobic, does not react with water. It just sits there. Does not interact with the water. So imagine from your belly up, you're hydrophilic, you like water, and from your belly down, your legs hate water. So now you dive in a swimming pool. Think about what might happen. Dive in the swimming pool, your head gets sucked in underwater and your legs get thrown out. And now you're like this little bobber bobbing up and down. If we go into the pool during gym class, we'd see a bunch of feet bobbing around and everybody's head stuck underwater, which wouldn't be good, obviously. And every time you try to pull your head up, you'd get sucked right back down in. Pull your head up, the water would pull your head right back down. And every time you try to put your feet down in the pool, they get rejected and thrown back out. Obviously, you're not built that way because that's not what happens to you. But part of you wants to be in the water and part of you doesn't want to be in the water. Probably beat yourself up taking a shower. So this molecule has to be, therefore, arranges itself in an interesting way. So over here, this is why that has to go into is arranged in what's called a bilayer. Here's layer number 1 right here. It's layer number 1. All these little gray balls represent the phosphates, and those are all the tails. Notice it's the phosphates that are in contact with the inside of the cell, which is water-based. So that's okay. They're all in contact with the water. But the tails are all pointing away from the inside of the cell, and they have no contact with the water. So they're happy. But if we only had one layer, the tails would be in contact with the outside of the cell, and that would not work. So we have to put in a second layer to protect the tails. So here are all the phosphate heads in the second layer. They're in contact with the outside water, and all their tails are stuck to the inside. So you have polar water-loving heads, and then an interior that's made up of completely water-hating tails. So if we label that, I don't think I gave you a box to label it with. So this creates a double layer. We call this a phospholipid bilayer. And so you've got, again, the hydrophobic heads. This box I don't think I gave you to fill in. So both regions that touch the inside and outside of the cell are hydrophilic, polar, but the inside is hydrophobic, nonpolar. And this is the key. This is what makes a chemical barrier. Most molecules cannot get through the tails. Most molecules will get chemically rejected by the tails and therefore cannot pass through the membrane. There's an old saying, like dissolves like. Like dissolves like. What that means is polar molecules will mix with other polar molecules, and nonpolar molecules will only mix with nonpolar molecules. If you try to put polar and nonpolar molecules together, they do not mix. There's a repulsion. Practical example, back 40, 50 years ago, if you were painting the walls in your house, almost everybody would have used an oil-based paint. Oil-based paints arguably are still a better paint than what we use today. They last longer, they go on better, they do better coverage. But the reason they're a pain in the butt, because they're oil-based, you can't clean the brushes and the rollers in your hands with water. If you try to clean the brushes, the rollers, or your hands with water, with oil-based paint, it doesn't do anything. The paint just stays there. The water just sits on top of the paint. They don't mix. You can't clean up your brushes and all that stuff with water. So it was not very convenient. So you had to use some highly flammable chemicals to clean your brushes and rollers. You have to use either gasoline or turpentine or something of that sort to do it. So along comes latex paint, which is all you guys ever known. Latex paint, the advanced latex paint, which is what you buy in the store now, is water-based, which means since the latex paint is water-based, you can clean up the rollers, the brushes, and your hands with soap and water. And thus, it's a lot more convenient and easy for people to use. So even though oil-based paint arguably is still a better paint, you get richer pigmentation, people don't want to go through the hassle of trying to clean up everything using gasoline or turpentine or acetone. People would much rather use soap and water and use the latex paint and get a similar look using that paint. Does that go anywhere? So like dissolves like, okay? So when it comes to the membrane, the only type of molecule that's going to get through here is things that are also nonpolar. If you're polar, you're going to get rejected by the interior of this membrane. It's not going to let you through. It's not going to let you through. That's what creates literally the barrier to things moving in and out of the cell. I have a good demonstration of this, but I can't do it online, unfortunately, to show you. But we'll do the best we can. So now the membrane also needs to be what we call semi-permeable. Semi-permeable means some things can pass through. Permeable means to go pass through. And some things cannot pass through. Okay? We can't just let anything go in and out of the cell anytime it wants. Then you have no control. So this membrane has to have some control as to what gets in and out of the cell to maintain the health of the cell, to maintain what we call homeostasis or balance. So it can't be a membrane that lets everything in. Otherwise, why have one? Again, we've used the analogy of the classroom walls being like a membrane. Well, if anybody can come in and out of the room and at any point that they want to, what's the point of having the walls? There's not much use to them. So the classroom has certain portions of the wall through which you can pass. We've put in doors and windows. And we have a little bit of control who goes through those doors and windows, so we have some control of what's happening in the room. Well, same thing with the membrane. It's got to be able to control what goes in and out of the cell. So the membrane ultimately needs to be able to move a number of things through. Okay, so semi-permeable, you're only going to move some materials can get in and out. Not everything can pass through. We don't want the membrane to be 100% permeable. So we need things to go across the membrane. We need things like sugar to be able to cross the membrane. And again, the blinking is not you. There's a glitch on my end. The lipids need to be able to pass through the membrane. Amino acids need to be able to pass through the membrane. Oxygen needs to be able to pass through the membrane. Water needs to be able to pass through the membrane. Salts need to be able to pass through the membrane. Waste products need to be able to pass through the membranes. All those things need to be able to pass through the membrane, but they all cannot do it directly because most of them are polar. Most of them are polar. They cannot get through the interior without getting rejected. If I end up going too fast, just let me know. So how do you get across the cell membrane? What molecules can get through the cell membrane directly? So which molecule, which molecule can start on one side and go all the way through the spaces to the other side? Which molecules can literally pass through these gaps and get through? Well, only molecules that can contend with this non-polar interior. So there's only one type of molecule can actually go through the cell. And those are fats and oils. Those are non-polar materials. A lipid can pass through no problem because it has the same chemistry as the fatty acid region. This is non-polar. It can pass through the non-polar region. But everything else that you need, what about the other stuff? Sugars can't pass through because they're polar. They can't go through the interior. Amino acids cannot pass through because they're polar. They can't go through the interior. Water can't, most of the water cannot go through the membrane because it can't go through the interior. It's polar. Salts can't get through. Waste products can't get through. So the phospholipid bilayer by itself pretty much blocks everything except for just a few handfuls of molecules that happen to be lipids. But every other type of molecule cannot get through that membrane. So there's your barrier. There's your barrier. But you still need those things. I still got to get sugar through. I still got to get amino acids through. I still got to get water through. I still got to get salt through. I still got to get waste products out. So there's more to the structure. There's more to the story. So what is another key component are what we call cell membrane channels. Sometimes you'll see them called integral proteins. Sometimes you'll see them called transmembrane proteins. Here we're calling them channel proteins. They are large proteins that are embedded between the phospholipid layer, between the phospholipid bilayer. So here they're showing you a very simple representation using colored cylinders. This is a yellow cylinder that will serve as a channel protein, this gray channel protein, this blue channel protein, pink channel protein, green channel protein. In reality, over here to the far right, this is a better representation of what that protein actually looks like. This whole folded protein chain. But we're keeping the picture simple by just making them cylinders. So you need to make doors through the membrane. If you want to get in sugar, you want to get in amino acids, you want to move water, you want to move waste products, you got to create doors for them to go through. You want to put walls to make a classroom and you want students to come in and out, you got to put a few doors in. So we got to create some doorways. And these channel proteins become those doorways. The channel proteins are what allow substances to go in and out and bypass that interior that is preventing them from getting in. And each of these channels is very specific. Each one of these is very specific. So we've got water channels that only move water. We got salt channels that only move salt. We got glucose channels that only move glucose. Different cell membranes have different channels. Kidney cells have lots of water channels because they filter the blood and produce the urine. But your skin cells have almost no water channels because we don't want you to dehydrate. Muscle cells have lots of glucose channels because they burn lots of fuel. Bone cells have very few glucose channels because they burn almost no energy. So one of the things that makes cells different is the different types of protein channels they have in their membranes. It varies from cell to cell. So if I want to move sugar across the membrane, we already illustrated that it can't go between here. The sugar can't get past those fatty acid tails. But if I use this transport protein, it can go through this protein without touching the fatty acid tails. You can't see it so well in this diagram, but there's actually a hollow space in here that the molecule passes through. It's called the channel. That's a channel protein. And that channel is polar, which allows the molecule to go through without interfering with the fatty acid tails. So if I need sugar, I'm going to move that sugar through the protein channel. Bingo, I've now passed it across the membrane. I can do that because I'm bypassing the fatty acid tails. If I want amino acids, I can move them across the membrane using an amino acid channel. If I want water, I can use a water channel called an aquaporin. Again, you can move both directions. If I want to get rid of extra salt, I need a salt channel to get rid of the extra salt. I've got a waste product like CO2 or urea. I can use a channel that's designed specifically for waste. So 5 of the major 6 molecules actually pass through the protein channels instead of through the membrane directly. And we've got 2 slides left here. So how do you build a semi-permeable cell membrane? How do you put one of these together? So you got channels which are made of proteins, but it can't be, so the problem is proteins are polar. So how do you stick a polar protein all the way through that phospholipid bilayer where the interior hates polar molecules? So what you have to do is you have to embed the protein, here's the protein, all the way through. Protein runs all the way through, but what happens is the outside of the protein here is non-polar. This part right here is non-polar, so it doesn't interfere with the fatty acid tails. Where the part that sticks up above and the part that sticks below, that's polar. And the interior is polar. So it's what's called amphipathic. You don't have to know the word. Think of amphibian. Amphibian means something that can live both on land and water. This is amphipathic, which means it has regions that can be both polar and non-polar, which allows you to embed the protein right through the membrane. So the membrane is primarily made of phospholipids and protein channels. Those are the 2 major components, the phospholipids and the protein channels. All right. So here we have the protein channels in the bilipid membrane. The only way this works is if this dark purple region here is non-polar, so it can compete with the fatty acid tails. It's okay if the outside's non-polar, but the inside, in order to do its job, has to be polar. And that, by that being polar, it can do its job. So they act like doors in the membrane. They allow specific molecules to move through the cell. This is a little bit more of a schematic drawing. So here's the more technical drawing right here. And this is a little bit more of a schematic drawing. So these proteins literally have a channel that run through them. You can't see the channel in this picture, but this is more of an idea. There's actually a channel that moves through them. And the molecules who cannot fit through here, they cannot go through this bilayer, these molecules can pass through the protein channel. They can travel through the protein channel. And that's how they get through the molecule. Okay. We'll get into this on Wednesday. You may already know there's a process called diffusion. And diffusion always moves molecules from high to low concentration. We'll go into why that happens and why it's always high to low. But diffusion for the body, for cells, is like free shipping. Everybody likes free shipping. So diffusion is a way to ship molecules at no cost of energy to the cell. But I'm getting ahead of myself. Okay. So again, proteins are going to tend to move molecules from high concentration to low concentration. All right. So for this moment, recap, cells need to constantly move materials in and constantly move materials out to keep the cell alive. What am I bringing in? I'm bringing in food, oxygen, water, salts. What am I getting rid of? Extra water, extra salts, dangerous waste products like CO2 and urea or ammonia. The membrane is structured to do this. There's a phospholipid bilayer, which creates a barrier to almost all molecules except lipids. And all the other molecules depend on proteins being embedded in that bilayer and allowing molecules to pass through the proteins. So some molecules, a small handful of molecules, can actually go right through the phospholipids. The majority of molecules are going to pass through those specialized channel proteins. And this gives the cell some control as to what goes in and out of the cell and how much goes in and out of the cell, so the cell can maintain a balance and stay healthy. Questions at this point? I know that's a lot of information. It's like, oh my God, 2 weeks off and you're giving me all this stuff. Can I smack my head? Anything? A little fuzziness would be understandable.
Did you
Go ahead.
put down, so on slide 4, where it says movement through the channel, did you get the second part of that, like, first page on the digital notebook?
You're missing a page, you said, or?
I just don't have where it says movement through the channel, those two boxes where you type.
Oh, on one slide, yeah. Don't worry. I did not have, I added those boxes after the fact, unfortunately. You can add your own box if you want, though. And I'll post the slides so you can see them. Anything else? All right. I'm going to go to the smartboard for a moment. What's my time at here? Holy crap. Time's flying. Okay. All right, I'm going to try to give you a little bit more of more detail on the membrane, just a little bit more. Then we'll take a look at this video, and then I'll set you loose on a task here. All right, so let me try this again. Should be coming. Beautiful. All right, I'm going to go for a walk here, just so I can get over by the smartboard. I can hear you. If you've got anything, I'm just going to be up at the board real quickly. We erase what's here and just restart. This is the one where you might want to make a drawing or just wait till it's up and take a screenshot or a photo. Either one is fine. I'm just going to add a little bit more detail to the structure, just a bit. So this is the cell membrane. Let's establish that this is the outside the cell. This is inside the cell. Just so we have an orientation. So we need a membrane to separate those two areas. So the basic component, again, are phospholipids. These phospholipids actually have small gaps between them. They're not super tight. There are small gaps between these phospholipids. I'm just going to randomly draw a few. And they have the two lipid tails. So the circles are the phosphate heads, and these are the two lipid tails. Because this is the outside of the cell. This is the outside of the cell. We have the heads facing the outside environment where it's water-based. But these tails are facing the inside, which is also water-based, so that doesn't work. So we need a second layer to protect the tails. So we're going to have a matching bottom layer with its tails facing inward, which solves the problem. Now, both water-based environments. The inside of the cell has got phosphates facing inward, and the tails are facing to the interior. So they're always in a bilayer because that protects the tails from becoming, they're always in a bilayer because that keeps the tails from coming in contact with water. All right? And so that's the structure. So these are the phospholipids. The second major component are the proteins, the channel proteins. And so every so often we have these channel proteins which are embedded in the membrane, and they go all the way through the cell, all the way through the membrane. And they create channels to move materials. So this label is a channel protein. This is also a channel protein. I'll just put CP, channel protein. And again, this part of the protein right here, these have to be nonpolar to be able to sit in the inside. This has to be nonpolar. These parts up here are going to be polar. It's going to be polar. That's going to be polar. The inside is going to be polar. And so we've got channel proteins. I'll introduce you to two other components that we're not going to spend a lot of time with right now, but we will in the future. There's two other major parts I think you should at least know about the membrane. One is cholesterol. Cholesterol is another lipid that's very important to cell membranes. Cholesterol gets a bad rap because, as many of you know, cholesterol can lead to heart attacks and strokes if you have too much cholesterol in your diet. But you do need cholesterol in order to maintain healthy membranes. You do need cholesterol to maintain healthy membranes. So cholesterol is periodically found in between the fatty acid tails. It's these green chains that I'm drawing in here, these chains of lipids that form cholesterol. Problem is, if you have too much cholesterol, it builds up in your blood vessels and blocks them. That's why it can lead to heart attacks and strokes. But under normal conditions, cholesterol are part of the cell membrane structure. And what the cholesterol does, it gets in between these fatty acid tails. Because what can happen is, particularly if the organism gets into a cold condition, the organism gets into a condition where the temperatures drop. Well, when temperatures drop, things condense. And the membrane would condense so tightly that there'd be no gaps or spaces and nothing could pass through the membrane. All of a sudden, the membrane would become impermeable. If the membrane becomes impermeable, you're going to kill the cell. So under cold conditions, you can't afford for the phospholipids to completely contract so tightly that there's no gaps. That's where the cholesterol comes in. The cholesterol gets in between those gaps, and it prevents the phospholipids from being able to contract too tightly. They'll contract to a point, but then the cholesterol prevents it from collapsing too tightly. It prevents it from becoming too tight. And it keeps the membrane, what we say, fluid. It keeps the membrane flexible. In order for membranes to transport material, they have to be fluid or flexible. Things have to be able to move around. So cholesterol protects the membrane, keeps the membrane from becoming too densely packed so that things can continue to transport through the membrane. So bottom line, the cholesterol keeps the membrane flexible and fluid, particularly under cold conditions. So we'll just label a couple of these. So that's cholesterol. Again, fluid and flexible. And the other one that I want you to at least have a little bit of knowledge of, and we'll deal with later in more detail, is on some of the lipids, you get these carbohydrates, and we call these oops, let me fix the spelling here. This one Come on, play nice. This one is called a glyco. The marker is possessed. This is called a glycolipid. A glyco is a glucose attached to a lipid, a phospholipid. That's a glycolipid. This is attached to the phosphate group, the phospholipid. Or you could also have them attached to the protein. If it's attached to the protein, we don't call it a glycolipid, we call it a glycoprotein. So they almost look like little antenna, and you only find these on the outer surface usually. They almost look like little antennae because what they are is they're receptor sites. Glycolipids and glycoproteins are receptor sites, and receptor sites are used for cell communication. Oh, it's really glitching. Anyway, there are these glycolipids and glycoproteins are used. I'll see if I can get the picture fixed in a second. These glycolipids and glycoproteins are used as receptor sites. They allow the cells to chemically talk to each other. We've already talked about why cells need to talk to each other. There has to be coordination between cells so that you don't damage something or damage the tissue. All right. So I'd like you to be familiar. I'm trying to get that picture to stabilize. I'd like you to be familiar with those four parts of the membrane. There's more to the membrane, but those four parts are the key part. Understand the role of the phospholipids and the proteins, which are the two major components and control most of the movement. Understand the role that cholesterol plays in keeping the membrane fluid and flexible. And for now, just understand that these glycoproteins and glycolipids help with cell communication. And we're going to get into cell communication in much more detail second semester. Let me try and see if I can bring this back up and see if it'll work a little bit better. Don't know why it's being so stubborn. If I can get up long enough, you can take a picture. So if you didn't get a chance to get a picture or to finish the sketch, it should be showing now at least. So again, the difference between those two blue structures is one's attached to a protein, that's a glycoprotein. One's attached to a lipid, that's a glycolipid. Essentially, they do the same job. Cell communication. Okay. Everybody good on that? That I can take it down? Okay. So let me present from over here. So let's go back to the notebook for a moment. Should be coming in just a second. All right, so on this page, there is a student reflection. So again, it's often good for learning to take a moment and stop and sort of process what you've heard so far. So what I'd like you to do is just think about what you've heard so far, and where it says student reflection here, you might want to change the font size. I think yours is going to come up at 18. You might want to drop it down a little bit. You don't have to, but might want to. I'd like you to come up with 3 or 4 things that you've heard today that are new to you and you think might be something worth knowing about cell membranes. All right? So just sort of look back at the other pages, think for a moment, come up with 3 or 4 statements about things you've learned about the cell membrane today that you think might be worth remembering. All right, so I'm going to be quiet for 3 or 4 minutes for you to fill in that page, and then we'll go forward from there. About 2 more minutes. Three or four things you've learned about how cell membrane is structured to transport materials. And at this point, it may not all make sense to you. That's okay. Just what do you think you understand so far? I'll ask you to wrap it up in about 45 seconds. If not, you can go back and add more to it. About 15. Okay. Again, you're welcome to go back and add more if I didn't give you enough time. Ultimately, you don't need to submit this until before you come to class on Wednesday. So next page, if you'll take a look at the next page, it is an Amoeba Sisters video, which I'm going to show now. And this one doesn't come with a handout, so I've just got a spot for you to take some keynotes on other information that you pick up about cell membranes here. All right. So I'm going to play the video. Yay. And you can either just listen and then add your notes afterwards, or you can add your notes as the video plays, up to you, whatever works for you. The video, as you can see, is about 9 minutes. And then following that, I'll give you the final task for Wednesday and let you go on your merry way till Wednesday. So have this page open. I'm going to go ahead and bring up the video. There is a link to the video right here as well, in case you need to go back to it, et cetera, et cetera. All right, so let me make sure I've got this. I just got to change the setting real quick. Come on, bugger. There we go. Nope. Okay. Should be coming back up in just a second. Just waiting for it to show up on the screen. There we go. All right.
In my second year of teaching, I was looking to improve an osmosis lab I had done the year previously.
Can you guys hear the
Osmosis,
video?
if you remember from
We can hear it. It just
involves
echoes.
water traveling
Yeah,
through
it
a
echoes.
semi-permeable membrane, potentially a cell membrane. And I wanted a cool way that students could model this in different scenarios. And one of my colleagues told me about this egg lab. I won't get into the whole lab, though. It was actually one of the first steps of the procedure that got to me. To get ready for the lab, my colleague told me, you can soak eggs in vinegar for 24 to 48 hours, and the shell comes off. Oh, so I need to make hard-boiled eggs then. No, raw eggs. But if the shell comes off, well, that's the whole point. What's underneath the shell is going to mimic a cell membrane. It's kind of modeling how a cell membrane would function. You know, if the
Did the
whole
echo
egg
go
was
away?
actually like a cell. So then you run different scenarios with it for your osmosis lab because it will be a semi-permeable
Yeah, the echo
membrane,
went
like
away.
a cell membrane. I couldn't visualize this. If the shell comes off, but it's a raw egg, how does it stay together? So I have this area in my house that's designated for things for me to try out. Don't worry, I always clean up afterwards. I tried this experiment out in advance, just to be sure. The hard shell is removed, but the membrane that was always there remains. We often visualize the membrane of a cell this way, like this membrane around the chicken egg. The cell membrane is semi-permeable, meaning it lets some materials through, but not others. We have an entire video all about cell transport and how materials can pass through the membrane. A cell could never be as large as a single chicken egg, though. Why? Well, it turns out surface area is a very important thing. Remember that surface area determines the surface measurements of that cell membrane, and the cell membrane controls what goes in and out of cells. That includes food coming in, as well as molecules that are essential for metabolic processes, and then also waste going out. If volume, which is all this space inside, increases, then you will have more need for surface area, as you will have more need for food to enter, more of a need for waste to be removed, and more metabolic reactions occurring in this larger volume in the first place. If we do a little bit of math here between these two models, I'm going to use popular cube models instead of egg-shaped models because it's a little faster for me to do surface area and volume calculations. See how here there is a big difference in surface area to volume in the smaller model? 6-to-1 ratio. That means the surface area in this small model is 6 times more than the volume. Look at this beautiful ratio with so much surface area. But if we...
It's echoing again.
Yeah, I'll fix it. This would make more sense because this is a lab we usually do in class. We would have actually probably done this lab today as our opening. So we may do this lab when you come back, for those that are coming back a little bit later on. But this is talking about why cells have to be small.
If we look at this bigger cube and we do some math for this model, that surface area to volume ratio decreases. Sure, the surface area is still larger than the volume in the large model, but it's only 2 times as large now, not 6 times as large. Cells are way smaller than this small model here to allow for an exceptionally large surface area to volume ratio. And a major reason why we're not going to find a cell as big as this chicken egg here. Surface area is important. And while we can model a lot of the processes of cell transport from this egg membrane and how important the membrane is, I don't want to neglect talking about the amazing cell membrane structure itself, because the cell membrane structure truly is magnificent. And since every single living thing is made up of one or more cells, which is part of the cell theory, it's a big deal because every single cell has a membrane. It doesn't matter whether you're talking about bacteria or protists or plants or animals or fungi. Even archaea aren't too cool to have a membrane. They all have a cell membrane. The structure can vary some, but we're going to be talking about some major structures of the membrane that you can actually find in most cells. We should mention that the fluid mosaic model is often what we use to describe the cell membrane. A mosaic, in case you've ever created one, we did in some of our art classes over time.
So this fluid mosaic model is how the membrane really works. The phospholipids and the proteins are not stationary. They slide around and they move around all the time. You can literally, if one part of the cell is getting more glucose than another part, you can literally move the glucose proteins over to where the glucose is to get it. So there's a lot of movement, a lot of fluidity in the cell membrane. It's called the fluid mosaic model, which they are trying to explain right now.
Arranges many small pieces together to make some large piece. You'll see that that makes sense when describing the membrane in a minute. The word fluid implies movement, and this is true for the cell membrane as the components are floating around, they're not static. So let's take a look at some of these components in the cell membrane. We're looking first at a phospholipid bilayer. A phospholipid is a lipid, but an interesting one. So when you talk about a lipid in general, many lipids are nonpolar. Think of oil, for example. It's nonpolar. It won't dissolve in water. Water is polar. But a phospholipid is interesting because one part of it is polar, the head, and the other part of it is nonpolar, the tail. It's amphiphilic. Let's explain what we mean. We often refer to the polar head of the phospholipid as hydrophilic, which means that part loves water. Well, you know, if a lipid could love. The nonpolar tails are hydrophobic. They do not like water. These phospholipids arrange themselves into a phospholipid bilayer with the nonpolar areas here in between, away from any water. It also allows this area in between to be separated from the outside and inside. Water can be found on the inside and outside areas. Also, these phospholipids, they don't just stay put. They move around. It's the fluid mosaic model after all. This gives the cell membrane flexibility. Phospholipids can even flip-flop around, but that's far less common. Remember that this entire phospholipid bilayer borders the whole cell. It would be a sphere, even though we're just looking at one area of it. We have an entire video that talks about which molecules can go in and out of this membrane and which ones can't that you can view, but for now, we're going to look at some of the other structures more in depth. Cholesterol. You know, cholesterol often gets a bad reputation. And while cholesterol that builds up in the arteries can be a problem, cholesterol in your cell membrane is critical. If temperatures drop, the cholesterol can actually function kind of like spacers between these phospholipids, keeping them from becoming too packed. Or vice versa, the cholesterol can actually function to connect phospholipids, to keep them from becoming too fluid in warm temperatures. Proteins. In protein synthesis, we talk about why it's so important for cells to make proteins. Many proteins are found in or on the cell membrane, and they play major roles. Peripheral proteins, like the name suggests, tend to be on the peripheral area of the membrane. So while they're on exterior areas of the membrane, they generally are not going to go through the membrane. That's for integral proteins. Integral proteins go through the membrane. Oh, and peripheral proteins can sit on them sometimes. Because of location, these proteins tend to have different functions. Integral proteins, with their potential to go through the membrane, are frequently involved in all kinds of transporting methods for all kinds of materials. Some relevance? Consider the breakfast you ate this morning. Your body digested what you ate for breakfast to obtain glucose. Once in the bloodstream, those glucose molecules can't just squeeze through the phospholipid bilayer to enter all of your cells. The glucose molecules are too big and polar. But your cells need glucose to survive, to make ATP, and they rely on integral proteins to get it. Peripheral proteins tend to be more loosely attached, since they're generally not stuck in the membrane. They have an assortment of functions such as acting as enzymes to speed up reactions or attaching to the cytoskeleton structures to help with cell shape. Both protein types can have carbohydrates bound to them, which can make them considered a glycoprotein. If the carbohydrates attach to a phospholipid, you could have what's called a glycolipid. Glycoproteins and glycolipids can identify the cell as belonging to the organism, self and non-self recognition, which is very important when you're fighting pathogens. They can also be involved in many kinds of cell signaling. In fact, here's some relevance. A glycoprotein known as CD4 is found on the surface of some of your immune cells. The CD4 glycoprotein is essential for some of these immune system cells to interact with each other and activate. However, it's also exploited by the HIV virus. The HIV virus uses that CD4 glycoprotein as a way to bind to helper T cells, which it then can infect. Understanding the components of the cell membrane and how those components are involved in recognition and cell signaling is critical to understanding how to fight back against many viral and bacterial diseases. Well, that's it for the Amoeba Sisters, and we remind you to stay curious.
Okay, so that gives a little bit of reinforcement to what I was trying to explain. The integral proteins, if you didn't catch on, integral proteins are the same as carrier proteins or channel proteins. The peripheral proteins I didn't specifically mention, but they do.
Michigan homeowners, if you have a power meter like this on the side of your house, you can qualify to get solar panels and erase your energy.
There we go. YouTube kept playing. Any particular questions? All right, so the intent is to go back again to the digital notebook and to make some notes or some observations. What new information came up in the film? What new insights did you gain by watching the film? Jot that down into the digital notebook. So once you've completed the reflection and you completed the notes from the video, you should be able to submit that digital notebook to Google Classroom. The other task that I'm going to ask you to do before coming to class on Wednesday is also under Google Classroom. It is the POGIL. Again, I initially was hoping we would be able to start that together in class, but that's not looking real probable. But this is the POGIL right here. It'll do a good job of talking about the cell membrane structure, but it's going to introduce you to the different types of transport that we are going to deal with in class on Wednesday. And there's a couple labs that we will set up as a demo because we're not in class together to do. We normally do them. You normally, as the students, do them. So one of the downsides of being virtual right now is we're missing a lot of the labs in person, but I'll at least give you a feel for what the lab looks like. So hopefully we're back on the 18th and we can start doing some in-person labs along the way as well. So before you come back on Wednesday, so we're all on the same page, before you come back on Wednesday, make sure you finish off the notebook. You should have it almost all done right now. Probably just had to maybe add a few notes about the video. Submit the notebook before coming back on Wednesday, and then complete this POGIL and submit it before coming back on Wednesday. Okay. Any questions? All right. I'm not going to hold you then. That's all I've got for today. You are free to go, and we will see everybody back on Wednesday. Enjoy the rest of your day.
Mr. Ed, have you seen the musical The Guy That Doesn't Like Musicals? Really? It's on YouTube. It's really good. I think we should do it. There's a little bit of profanity, but I think we can change that out. Make it a little more appropriate for high school.
The guy who doesn't like musicals?
Oh my gosh.
It's so awesome.
I'm gonna have to check that out later then.
Yeah, so basically, the basic thing is his town, a meteor hits, and it's a magical meteor that makes the entire town a musical, and it's his own personal like hell, and he's like, why is this happening? And he's like running around, and then he finds someone else that wasn't infected by it, and then they're all like trying to make it out because like they became musical loving aliens, and they're all doing like choreographed dances and stuff, and he's like, ah, it's awesome.
I don't even know anymore.
All
right, bye.
Well, I got marked absent.
Because I have to take it right at the beginning, and I kept asking Drake, "Are you here? Are you here?" and nobody responded.
Oh, sorry, my thing wasn't working.
Right, but I will. I saw you come in, so I will fix it.
All right, thank you.